see you there. I was enjoying some public school law. But guess what time it is? It's time to biologize.
Nobody's heard from me for months. I'm doing better than.
All right, so test number 4, Unit 4 test. Talking about DNA replication, and mitosis, the cell cycle, how cells make more cells. So let's kind of go through some things first, and then we'll look at the just some kind of things to know. So remember DNA, okay, deoxyribonucleic acid. Okay, so DNA is what gives every living thing their traits. Every living organism has this genetic information in the form of DNA. And we talked about DNA being built of these, I said in class, these chunks of DNA, right? And they're called nucleotides. And we said nucleotides have 3 parts. Nucleotides have a phosphate, they have a sugar called deoxyribose, and then the most important part is going to be the nitrogen base. And so if we think of DNA as this ladder, and in fact we say this twisted ladder, the fancy name for it that Watson and Crick found was a double helix, right? So the sides of the ladder would be made of the phosphates and the sugars. The middle rungs of the ladder then would be the nitrogen bases. And remember we said the nitrogen bases are the most important part because the order of those bases is what gives you and all living things their traits, okay? So the nitrogen bases were specific, right? There was adenine, thymine, guanine, and cytosine. So we had A, T, G, and C. And we talked about this idea of these base pairing rules, that these bases are attracted to each other, right? In a certain way. So we said apple in the tree. So A's and T's always go together. And then we said car in the garage. C's and G's always go together. And why is that helpful? Well, the next part of the whole puzzle was we talked about DNA replication. So before our cells can make any copies and make any new cells, there has to be a copy of the DNA in each new cell. So to do that, DNA has to replicate. DNA replication happens in 3 steps. The first step we said was the double helix has to unwind and unzip, okay? So once it does that, then it's just like a zipper, it unzips, and you've got the 2 parent strands, right? You have the 2 original strands. Second step then is these free-floating nucleotides start to attach to each side, okay, each side of the parent strand. So the A's match up with T's, the T's match up with A's, G's to C's, C to G, and at the end of it then what you get is 2 identical strands of DNA, okay? We said that DNA replication is something called semi-conservative because the original strands, once they separate from each other, you're using those strands as what we call a template strand. We also say, again, like those are the parent strands, right? The original strands that we're copying off of, okay? To then, again, what's the ultimate goal? 2 identical strands of DNA. So that was pretty much our DNA. The other thing we need to make sure we understand, the order of the nitrogen bases makes you different than the person sitting next to you. But we also said the order of the nucleotides makes you different. Why is that kind of saying the same thing? Because remember, the nitrogen base is a part of the nucleotide. And I said today in class, remember at my little conference I was at, we said the little hand motion. So we've got our sugar is the fist, the phosphate's the thumb, and then the pinky would be the nitrogen bases. So again, the sides of the ladder here would be the phosphate and sugar. Then we got your nitrogen base right there. And then there's your double strand, okay? And again, if I move this whole chunk, if I move this whole nucleotide, it's rearranging the base too, right? And so that's why, the order of the nucleotides, the order of the nitrogen bases, that's like saying the same thing, okay? The second thing we looked at was mitosis and the cell cycle, right? So the cell cycle is kind of the whole life cycle of a cell. And we said that There's three things. There's interphase, mitosis, and then cytokinesis. So interphase, you have three parts. You have G1, where the cell's growing. You have the S phase, that's where the DNA replicates. S stands for synthesis. So remember, we're synthesizing the DNA, right? And then the last part of interphase is G2. That's where the organelles kind of replicate. Okay, and again, there's a little bit more growth. The M, or the mitosis part, comes next. And I know we did that whole pie chart, right? So most of the time the cell is in interphase, and then mitosis comes. So there was 4 stages: prophase, metaphase, anaphase, telophase. So let's go through those real quick. And this should all be in your notes. Prophase, remember, the nucleus disappears. The chromatin, the tangly thread-like stuff, condenses into chromosomes. Then secondly, we have metaphase. Metaphase, middle, right? So the chromosomes line up in the middle. Next comes anaphase. Anaphase, away. So the chromosomes, the pieces of the chromosomes, move away from each other to opposite ends of the cell. That's important so that there's an equal amount of genetic material inside each new daughter cell that's going to be formed. Then we have telophase. Telophase, you have the 2 new nuclei formed, and then the chromosomes uncoil and go back into chromatin, the real thread-like stuff. And then finally, cytokinesis splits the cell into 2, and you get your 2 identical daughter cells. So we said in the notes, you know, when does this happen, or what kind of cells does this happen to? Remember, it's our body cells. All of our body cells are what would go through mitosis. Body cells, the fancy word for body cells is something called somatic, our somatic cells. Our gametes, those are our sex cells. So our gametes are the sex cells that there's a whole different process that we're going to talk about later on how those replicate. But our sex cells do not use mitosis. Only our body cells do, or our somatic cells. Okay, so a great example of when mitosis or the cell cycle would be happening is when, you know, you get an injury. Maybe an animal gets a cut or something, or you get a cut. Mitosis is going to take over to try and seal that up real quick. Okay. Then we talked about cancer, right? And you're doing this whole project with cancer, and we said the big thing with cancer is, or tumors or any of that stuff, is when the cell cycle goes out of control. So there's these checkpoints that happen during interphase, and if they get screwed up, or if there's something wrong with the DNA and the cell continues on into mitosis and keeps replicating over and over and over, that is when cancer and tumors can form. Cancer is basically a cell that's lost its ability to control the cell cycle. That's when tumors and cancer start to form. So keep that in mind. There's definitely a couple of questions that have to do with that. Now let's see, we can look at the test a little bit and see what we got here. Again, what's the end result of DNA replication? Remember, you want to have 2 identical daughter cells. There's one talking about tumors and why they're growing. It's pretty much what I just told you. Just a tip, a zygote, it would be like the first cell of when a sperm and egg come together. That would be a zygote. Just as a term that maybe we haven't used a lot, but it's just so you'll see what I'm talking about. Remember, pictures of anaphase, you'd have chromosomes separating, right? Going away from each other. If there was a picture of metaphase, you'd have chromosomes lined up in the middle. So make sure you're able to identify pictures of the stages, mainly anaphase and metaphase, but make sure you are thinking about what the chromosomes are doing, okay? What happens during S phase, right? replication. Way to go. Let's see. How many cells do you get at the end of mitosis? Remember, we get 2 identical daughter cells. So you get 2 identical cells. If you see any kind of picture where there's 2 strands of DNA, what do you think's happening? Replication, right? If it's showing you 2 strands of DNA, replication. Remember we talked about the parent strand, how the parent strand, kind of guides the 2 new strands being made. Okay. Remember, nitrogen bases is where all the traits are actually held. Remember your base pairing rules. So A's to T's, G's to C's. If you had a segment of DNA, you need to be able to fill in the opposite side. Okay. Remember, there's always going to be an equal amount of A's to T's and C's to G's. You had several questions kind of leading up to this about the percentages, right? So you need to be able to figure that out. What do we call, what's the fancy name of the DNA molecule? Remember we said it's the double helix, right? Okay. What else? Oh, is it dinner time?
Wash your hands for dinner time.
Wash your hands for dinner time. Oh, thanks, Carlin.
Mom told me to do it.
So again, 2 strands of DNA, you'd have replication. That's kind of a no-brainer there. Let's see here. Oh, what are the nitrogen bases held together by? Remember we talked about that. So when you make your little pinkies here, they're held together by those hydrogen bonds, right? And that's what breaks apart so that they can replicate. Oh, the cameraman's getting tired. Let's see. I think that's about it. Remember, all organisms share genetic material, right? They all have DNA. It's not going to be the exact same sequence, right? That's what makes us different, but all organisms have DNA. I think that's about it. Again, I'll be there in both tomorrow morning and Wednesday morning. If I think of some other things, I'll add another clip here in a little bit, but that should be about it. Watch the other lectures that are on YouTube. Make sure you check those out. And please make sure you're studying. Don't forget about the contest. Let's take it, right? Competition. Let's do it. Cameraman, stop dropping my phone. See y'all later. Shout out to Colt, the cameraman. Yay. Yeah. There